Today we are with Fernando Vieira with ALS TDI, and we're hearing about their exciting research they're doing with your donations to Racing for ALS. My name's Scott Weatherford. I've been involved with Racing for ALS since the beginning because my friend David Lloyd was diagnosed with ALS, and I saw the symptoms progress from taking him off the racetrack to not even being able to participate in this video today. So our donors, our corporate sponsors, our social media followers would love to hear the great work that ALS TDI is doing, Fernando. So thank you very much for joining us today.
Thank you for having me here, Scott. It's a pleasure.
What does ARC stand for again?
It's the ALS Research Collaborative.
Okay.
It's a program where we're really partnering directly with people with ALS to learn about the disease directly from them. And yeah, I'm excited to tell you about it. ALS Research Collaborative, or ARC, it's a program that's designed to learn about the disease from people with it. We've been also collaborating directly with a lot of researchers around the world to really accelerate the research that'll be needed to find effective treatments for the disease. I'm going to talk to you a little bit today about some of the reasons why it's hard to find treatments for ALS, but why a program like this can help us make the essential progress. One of the things that we talk about in ALS is this word called heterogeneity. What we've been realizing in ALS over the past few years is that it's probably not just one disease. It's probably a collection of different diseases that all result in the same problem, where your motor neurons, the neurons that control your ability to move your voluntary muscles, fail and degenerate and die. So it makes it really hard for people to move. We're learning more and more that there might be lots of ways to get there. And that shows up a few different ways. So I have this figure that I wanted to show you from a paper from a few years ago, where they show that different manifestations of ALS, different ways that the disease starts, your earliest symptoms, can kind of predict how the disease is going to go, whether it's going to be faster or slower in you. So what you're seeing is that, like here, if you look at this yellow line here, you're looking at a population of people represented in that line, and they have a certain type of symptom. So all of their symptoms are tied to problems with the neurons that are in their brain, not the ones in their spinal cord. You have motor neurons in your brain and in your spinal cord. If your disease starts where it's mostly the neurons in your brain that are affected, you tend to live longer with the disease. Whereas somebody whose first symptoms are the ones that affect their breathing, their respiratory, you tend to live a shorter amount of time with the disease. And we don't understand why this is yet. And it's going to be important to understand the why of that, so we can understand a lot of the how we can change it and make it better for everybody.
This is a very powerful graph, and that's seeing the data laid out like this just reinforces the importance of the work that ALS TDI is doing.
Thank you. It is. And you have to be mindful when you're looking at graphs like this that we're looking at information about people. These are specific people, right? It's not just data. And if you've been in the ALS space like I have, I lost my best friend to the disease. It hits different. It makes an impact. And so thank you for pointing that out. So I talked through how ALS can be different in terms of your initial symptoms and how it can progress differently that way. We're also learning at the other end of the spectrum, at the molecular level, sort of the building blocks of a person, at the genes. We're seeing that there are a lot of different genes that can be mutated or broken that might tip a person to eventually develop ALS. So each one of these dots here represents a different gene, which was identified as a risk factor for ALS over all of these years. You can see the field, we're making a lot of progress. We're discovering a lot of these different initial dominoes that can be tipped, but they all have different jobs. So this also tells us there's heterogeneity. There's that word again. There are a lot of different ways that you can get ALS. And we're going to need to understand those different ways to find the different ways to treat it.
The complexity of the disease, right? That just reinforces how challenging this is, and that it's not going to be a single magic bullet that's going to solve this problem, and that it just reinforces how challenging this is for ALS TDI and other researchers in the field.
Absolutely. It's going to take a lot of magic bullets, right, to get there. And I'd love to kind of talk a little bit more about how I think we can get there. So, and that's our job, right? Our job is to, in my mind, fill this graph. So I'm going to explain this graph to you. So on this axis, what the ideal would be to have a treatment or a collection of treatments that result in maximum efficacy. So 100% efficacy would be a treatment that stops a person from ever having ALS or completely reverses their disease so they live a normal, long life with full function. And on this line, on this axis, is the number of people. So you would want, ideally, to accomplish that for everybody. So our job, in my mind, is to fill this graph. We're a little bit of a ways off from that right now. The status right now is something a little bit more like this. We've got some treatments that are marginally effective, that help a little bit with a lot of the people, but not everybody, right? So they're making a little bit of a difference, but we're not nearly where we want to be. Recently, because of a better understanding of a specific subtype of the disease, a specific mutant, specific gene, a group was able to invent a new treatment that was a lot more effective for those folks. So that's what you're seeing here with this bar. We're seeing more efficacy, small population. What we're going to have to do is stack treatments on top of treatments until we completely fill this rectangle, right? So we're still not there with this subset. We've made an impact, but we're still not fully where we need to be. We got to fill that in. And then there may be some types that the current treatments don't help at all. And we're going to have to understand those subtypes as well until we fill this all in. And that's our job. So how do we get there? So there was a international group of ALS researchers. We all convened near the end of 2022 to kind of talk this through. Like, what are some of the things that are going to be essential for this? And what was clear is that this heterogeneity is a big challenge, both from how the disease presents at its symptom level and the genes that can go wrong in it. And to really understand it, we need to have a large study that tracks lots of different people with ALS over a long time and measures symptoms a lot of different ways. And that's essentially what this is. So when you hear natural history study, that's what we're talking about. It's a study that tracks people with the disease over a long time so that we can better understand it. So the ALS Research Collaborative, we actually initiated this. We recognized this need back in 2014, and we started enrolling folks with ALS back then, recognizing that we were going to really need to get kind of a complete picture of lots of different types of people with ALS to understand it. So we're going to track how their disease progresses. We're going to measure a bunch of things, a bunch of the biology in their blood, and that's what we call molecular omics. So you've heard maybe genomics, but there are other omics. There's transcriptomics, that's measuring RNA. There's proteomics, that's measuring proteins. There are lots of different omics. And then there are different biomarkers. If you don't know what a biomarker is, it kind of says it in the name. It's a marker that you can measure. It's a way to measure how your disease is progressing. If you can measure how it's progressing, you can measure if you're fixing it, right?
For example, that's some of the voice analysis work that you're doing with ALS patients.
Exactly. So when we started this program, Scott, we said we're going to collect as much information as we can, even if we don't know what we're ever going to do with it. So we collected a lot of voice recordings. We've now collected more than 10,000 voice recordings from people with ALS. Because if you don't know, a person's speech in ALS can be very affected as the muscles in their mouth and throat and diaphragm start to fail. We're also using,
and that's actually why David isn't here with us today, is because he's having a bad speech week.
I'm sorry to hear that.
Yeah.
It's one of those types of things that you can qualitatively know, right, that a person's voice is getting worse. But how do you put a number on that? And how do you measure if a drug is making it better, right? That was a gap. And so we decided that if we collected these data, we might be able to find a way to measure how to make that better over time. And I can talk to you a little bit more about that as we go along. But so to date, we've now enrolled more than 800 people in this program. It's an open science program. So that means that that's part of why we're calling it the ALS Research Collaborative, because it's a giant collaborative effort, not just directly with people with the disease, also with other researchers. So any researcher around the world can access the data that we've collected. And I'll tell you more about that as well. And like I said, our participants, the patients, are our partners. We share the data back with them too. So a person can log into their own portal, like it's sort of like, I don't know, maybe if you have like a Facebook account or a LinkedIn, you log in with a username and password and see your own information. You can do that if you're part of the ALS Research Collaborative as well. And that's really important because it's their data. It's not ours. Each participant, it's their data. So we're collecting information about, yes, how their disease progresses. I touched on it a little bit already in terms of speech and other things, but we're also looking at survey data. So a lot of how ALS progression is measured is asking about your ability to do things in your daily life. Can a person, walk up the stairs? Can they turn themselves over in bed? Are they able to cut their own food?
One of the things that stuck with me is one of the people that we've interfaced with. Her thing was tying her shoes.
Yeah.
And she tried, struggled, and got her shoes tied eventually, but that was a win for her that day, and that really stuck with me.
Yeah, so we're trying to capture that type of information. It's hard to put a number on that, right? And so a lot of the things that we're trying to do is find ways to put a number on that type of skill. So again, you can measure the day that you're doing better, and if that might come because of a treatment. So this is a major emphasis here, is measuring symptoms and progression. We're also measuring risk factors, or asking about risk factors, really. So, there are a lot of different ideas or hypotheses for what might cause ALS. We know that genetics are involved. We've talked about that. But not everybody who has a genetic risk factor gets the disease. So what else might there be? Could there possibly be things in your life that you did or were exposed to that might have triggered it. That's not the massive emphasis of what we're doing. We're more interested in trying to intervene for people who have ALS now, invent treatments for folks living with the disease. But if we can learn ways to avoid developing it, we want to do that too, and we want to share that information. And then we do a whole lot by measuring thousands, if not millions, of things in the blood. Your genome is a little bit like a very, very long book with plans for how your body is going to build itself. And it's we're reading every single letter in that book and putting it in order. That's whole genome sequencing. So we're doing these surveys to track disease. We're using like accelerometers, which are a lot like Fitbits on steroids, where we're tracking a person's ability to move each one of their arms and each one of their legs. And we're measuring if there are differences early in disease and how it progresses through life. And then again, like we said, with speech. And we're partnering with groups like Google and other researchers over at Massachusetts General Hospital to use artificial intelligence and machine learning to put a number on these things. So if a person has normal speech, yep, you get a score of a 4, right? You would probably score as a 4. I would probably score as a 4. If I have a cold, maybe I score as like a 3.8, something like that. Our ability to take a voice recording from a person with ALS and run it through a computer and a machine learning algorithm for it to tell us, oh, this voice is at a 2.2 today. Yesterday it was a 2.3, or vice versa. So it's completely innovative. Nobody's really ever done this before. We're really excited to have partnered with Google to make that happen. And we shared those data and that algorithm with the world already.
Machine learning and AI and just the computational power and the amount of data that y'all are collecting, it would be impossible for independent researchers or individual researchers to use this data in a meaningful way. So it's really amazing to see the combination of technology and medicine come together like this in order to solve this problem that's so vexing.
People ask me, I've been working in ALS for more than 20 years. And people ask me, how do I feel about it, after all this time? And it's incredibly exciting because of the technologies that we can bring to bear today that weren't there 2 decades ago. There's so much more potential, but we've got to do the job of catalyzing that, of like using all of that tech. Sometimes that's ourselves doing it here, and sometimes it's finding the right folks to help us do it, and sometimes it's letting folks find us and then go do something independently, right? With the data that we can share. That's the power of today, essentially. What's next for ARC? Well, I told you that we've enrolled to date more than 800 people. We intend to grow it to 2,000 people, right? We really want to get it to 2,000 people as quickly as we possibly can. We're going to collect more blood samples for more biomarker discovery to really understand what's happening in the biology. If we can understand, for example, Scott, what makes a person's ALS go slowly, right? We've got some people who have slower disease than others. If we can understand how, and we can replicate that in everybody, then maybe everybody can have slow ALS until eventually we can have everybody having reversed ALS, right? So we're going to try to learn more about the disease from folks with it and see if we can use that to guide the discovery that happens in our labs right here in Massachusetts. And then one other thing that I haven't talked about yet is that we're adding electronic medical records, right? So everybody who's in the medical system in the United States, at least today, now has an electronic medical record. And those records go back to before you ever had ALS, right? Those records go back in your life, and that can teach us more about what might have driven the disease in your life. Maybe we can find out that there's a type of infection that a lot of folks had in their 20s that might have triggered a disease. Or maybe we'll find out that, yeah, it's folks who've had a lot of concussions, right? We've learned about some of these risk factors, but if we can integrate that into the rest of this program, it can be incredibly powerful. So we're excited about where the program is going next. And all of this, like I mentioned, is now shared with researchers around the world through our ARC Data Commons. And this is all using Google Cloud and Google's Looker interface, which allows people to kind of filter through, sift through, visualize the data incredibly quickly. We make the data available to academic researchers and nonprofit researchers everywhere at no cost. But that does come at a cost to us. So none of this entire program, the data collected and the data shared, none of it would be possible without support from folks like Racing for ALS, who really make it possible. So I want to make sure all of your entire community knows how grateful I am and how grateful everyone at ALS TDI is for that support.
Well, thank you for doing the work, right? And, ALS TDI has been a leader in ALS research. And, ultimately, we're asking for donations and sponsorships and doing our events to help you find a breakthrough, to have a eureka where we can end ALS.
Yeah, we're on our way. You know what? I bet we've already had a few eureka moments. It takes a while for it to play out, to get to that clinic and to prove that your drug worked. But we've got some stuff in the pipeline, and I'm really excited to see where it goes. More support, more help, and we'll get there for everyone.
Well, ALS TDI is a major part of the Racing for ALS Triple Target Initiative. And, it's really great to see the progress that you're making.
Thank you, Scott. And thank you to all of your viewers at home.
Well, thank you very much for watching the video. If you want to learn more, ALS has a great website, als.net, and you can even learn more about the ARC project. And please visit racingforals.com to learn more about the charity and our events. So, Fernando, thank you very much for all your time. And this is really exciting, and we're really hopeful it leads to a breakthrough.
Thank you. Looking forward to it.